Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nano Alerta. Hoje, no seu número 57. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia no Brasil. Também fazemos este programa porque é aqui que nós podemos discutir os impactos da nanotecnologia no meio ambiente, na saúde, na economia, na sociedade, coisas que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme nós tínhamos anunciado, já estamos conectados com a cidade de Chicago, nos Estados Unidos, onde estão o professor Reinhold e o professor Larson, que vão nos falar hoje de um importante tema da nanotecnologia com quem eles têm trabalhado. Primeiro eles vão se apresentar e depois nós iniciaremos a apresentação deles programada para hoje. Ok, Reinhold, is with you.
Bom dia, Paulo.
Bom dia.
Thank you very much for having Jeremy Larson and me on your internet television program.
Okay, you can speak about your experience, professional experience, and with nanotechnology.
Very good. I am a professor at the University of Wisconsin-Milwaukee since 1986. I received my bachelor's and master's degree in Chicago, Loyola University of Chicago in biology, biología, and then at Michigan State University, PhD in physiology. And then I went to the University of Wisconsin-Madison for postdoctoral research in reproductive endocrinology. And I was using various animal models like monkeys to look at the effects of estrogen and molecules like estrogen to control reproductive function and to understand how estrogen molecules work in the regulation of the ovary and ovarian follicle development. So then I came to Milwaukee, to the University of Wisconsin, and we were continuing our work in reproductive biology, and we began to look at other molecules that can affect the way estrogen does its work. We call it the estrogen receptor signaling pathway. And so we looked at different molecules like polychlorinated biphenyls, PCBs. Very recently, we've looked at bisphenol A, for example. But then about in 1994, we came across dioxin, TCDD, which is one of the most poisonous molecules on the planet known to man. And but however, this molecule has anti-estrogen-like effects. So we were interested in using the molecule in our model of controlling reproductive function. And so then we started working on dioxin in toxicology. And so from that point on, we were doing many different toxicants. And then about 4 or 5 years ago, I had a graduate student, master student, Rosemary Weber, who I gave a project to on nanoparticles. And I will show you some of that information on the slide presentation, the PowerPoint. But so we were interested in looking at the cells in the ovary called granulosa cells and whether gold nanoparticles that are very small, about 7 to 10 nanometers in diameter, can cross the cells and have an effect. she did work using electron microscopy and immunoassay, and we found that the nanoparticles changed the ability of the granulosa cells in the ovary to produce estrogen and some other molecules also, other sex steroids. And they also were found under electron microscopy to go through the cell membrane, enter the cell, and then enter some organelles that are involved in steroid synthesis, like the mitochondrion and also lipid droplets and other molecules. And then much more recently, a few years ago, Mr. Jeremy Larson came into the lab. We were fortunate to have him as a graduate student, master's student, and he developed a model using the rat where he took the ovaries and put them in culture in vitro, characterized the gold nanoparticles significantly. We collaborated with physicists and chemists and other people. He will talk to you about that also. And we were able to discover that there are some subtle changes in the ovary and effects on some of the enzyme genes that are involved in producing sex steroids like estrogen, progesterone, androgens. So I will then now let Mr. Jeremy Larson say a few things about his history and about his project.
Okay.
Hello.
My name is Mr. Jeremy Larson, and I'm a former student in Reinhold Hutz's lab. I graduated in 2008 with bachelor degrees in biochemistry, human biology, and psychology. And in 2012, I graduated from Reinhold Hutz's lab with a master's with focus in nanotoxicology. And I developed the rat ovary model focused on looking at the effects of gold nanoparticles on estrogen and progesterone production, as well as some of the genes in the steroid pathway.
Okay, very good. We can start the presentation now.
Very good. First slide, please.
Is the title, is in the screen.
Yes. Oh. Okay, so the title of this talk is Novel Environmental Endocrine Disruptors of the Ovary. We have been all along, as I mentioned, looking at the effects of various molecules and how they could affect the ovary and the production of estrogen and other molecules by the ovary. Molecules that can affect hormone systems are called endocrine disruptors. And so the terminology has been here since the 1990s or so. So as I mentioned previously, we've looked at molecules like PCBs and dioxins and other molecules, but we are now pursuing the possibility that gold nanoparticles and other nanoparticles are novel or new environmental endocrine disruptors of the ovary. So if we could have the next slide, please.
Não, aí, não sei. Okay.
So these are some of the institutions that we are working with. The very top, you see that we are at the University of Wisconsin-Milwaukee, but if below that, you will see that we have collaborations using monkey tissues from the Wisconsin National Primate Research Center. To the right of that, I have a affiliation also with the Medical College of Wisconsin, departments of physiology, pharmacology and toxicology, and obstetrics and gynecology. So we have received human ovarian tissues and cells from women that are participating in vitro fertilization programs. And then to the right, here at the University of Wisconsin-Milwaukee, and also at the Medical College of Wisconsin and the Children's Research Institute in Milwaukee, we have a center called the Children's Environmental Health Sciences Core Center, and we have collaborations there and use some of their services. I also have a connection with what is called the Great Lakes Water Institute, and we have a brand new School of Freshwater Sciences, which is the only one in the United States, and there are only a few worldwide. And so we also do work in zebrafish and other fish models looking at the effects of nanoparticles and some of these other toxicants. Next slide, please.
Okay.
So what are nanoparticles? I will say a few things, and then Mr. Larson will run in. He is an expert on some of the characterization of these nanoparticles. But these are particles that are obviously very small in the nano range, and so we say that in one of their dimensions, they're between 1 and 100 nanometers. So this very small size confers various unique size-related properties. And so, for example, the spheres, if we're working with spheres, have an increased surface area and some other things that Mr. Larson will mention now.
Okay. The small size of the nanoparticles confer us similar unique properties based on that size, such as surface area, and this increased surface area increases the chemical reactivity that can potentiate toxic effects in cells. And also you have the surface chemistry, different molecules that are either intentionally or unintentionally attached to the surface of the nanoparticle that can affect its reactivity.
No.
And it also affects the solubility of the particles, whether they are in solution or not, as well as diverse shapes and sizes. Typically, the smaller the particle, the more reactive due to an increased surface area. And different sizes can confer different reactivity within the cell as well. Next slide, please.
Yes.
And the nanoparticles come in various shapes, forms, and compositions. So you have the quantum dots that have a semiconductive property. You also have the dendrimers that are mostly organic molecules, and silica, carbon nanotubes, oxides, metals, and you can see there in that image that there's a variety and a high diversity in the shapes and sizes that can have a lot of different applications, both positive and negative effects.
I can add a few things here. If you look at the fourth bullet point, Jeremy had mentioned carbon nanotubes, but also DEPs, or diesel exhaust particles, have been shown to have various effects. We collaborate with Dr. Gen Watanabe at the Tokyo University of Agriculture and Technology and his team. I've been collaborating with them since the mid-1990s or so. And they have in recent years been looking at particulate matter in diesel exhaust DEPs, or PM2.5, for example, which are 2.5 microns in diameter. But in diesel exhaust, there are also ultrafine particles. These are less than 100 nanometers, so they can also be characterized as nanoparticles. And his laboratory group has shown that these DEPs, using a male rat model, can have effects on luteinizing hormone LH and affect the testosterone output of the testis in his model. And then toward the bottom of that slide are the metals that we've mentioned. So some people work with silver nanoparticles. We are working with gold nanoparticles for certain reasons. And then other nanoparticles are also buckyballs, or C60, C80 molecules. And there are other types that fit in that category also. Next slide, please.
Yes.
We have listed a few comments about the use of nanoparticles and our exposure naturally on a day-to-day basis. Nanoparticles are ubiquitous. They are found everywhere. So they're being used in research in medicine, electronics. The cosmetics industry uses them a lot. Many other consumer products, we will show you some of those slides coming up. And so we are highly exposed to nanoparticles in the environment. And at the bottom there it says it's estimated that 10 to the 12th to 10 to the 14th nanoparticles. So we have a huge number are ingested, so taken orally per person per day in the Western world. But unfortunately, we still do not know if these particles have a negative or detrimental effect on us, and that's what we are interested in a toxicologic way. Next slide, please.
Yes.
There are various types of exposures. That is how we are exposed to these particles. The most likely exposure is accidental or involuntary, and these exposures can occur through the skin, orally, taking them, ingesting, breathing them in, so via the lungs, and the nanoparticles can show rapid movement through the bloodstream to other organs.
And I'd also like to add that the route of exposure can be a detrimental factor in the toxicological response. Different routes of exposure will exert a different toxicological response. So really, that is really a key determinant or factor that has to be taken into consideration. If it's skin absorption or orally or via injection, that route of exposure will expose the nanoparticle to a specific suite of proteins and molecules that will affect its behavior in vivo.
So we will see that each nanoparticle is completely different depending upon its physical characteristics, and then how one administers it or how one is exposed to it can completely alter the biologic effects that that particle can have. So certainly not all nanoparticles can be treated the same way. They are all distinct, completely different. Even the exact same type of gold nanoparticle, if the size is different or if the surface is functionalized or has other molecules on it, can totally affect the way that molecule works. Next slide, please.
Yes.
Okay, here this slide shows a lot of the consumer products that have been shown to have nanoparticles as a part of them. From the Comfortone, your typical suntan lotion will have titanium dioxide or titanium oxide nanoparticles. You also have a lot of electronics, such as TVs, computers, that use various types of these molecules, tires, cosmetics, sports equipment, as well as waterproof clothing, antibiotic resistance in socks and other undergarments. There's a lot of everyday products that we take for granted that have nanoparticles as a part of their structure.
We have recently seen on the web, for example, advertisements selling gold nanoparticles within toothbrushes and toothpastes. I have not seen that in any stores here that I have been to, but that is supposed to take advantage of bactericidal effects, killing of bacteria on toothpaste, toothbrushes, for example. Next slide, please.
Yes.
Okay, and this slide as well shows that there are a lot of different applications of nanoparticles. Like I said, with the suntan lotion, socks, specifically silver nanoparticles, have an antimicrobial property that is often used to keep our socks smelling clean. You also have light-emitting diodes, scratch-proof coated windows, fabrics coated for resistant to stains, nanoparticle paint to prevent corrosion, magnetic layers to compact data memory, just various applications that can be beneficial to our society and to our world, but we just don't know what harmful effects that they might have on human health.
There's an interesting aside. We have written there below the socks diagram, antimicrobial socks that deodorize. At least some of those silver nanoparticle socks, once they are washed, they typically lose their nanoparticles so that the socks no longer have that antimicrobial effect, certainly after a few washings. Interesting aside. Next slide, please.
Yes.
And this slide as well shows, a great kind of emphasizes the great diversity in the application of the nanoparticles, from textiles to drug delivery to different applications in healthcare, agricultural, meat, industry, electronics, renewable energy, fuel cells. There's another hot topic of research in nanopesticides. And nanopesticides are believed to be more effective because they can have a higher reactivity with the lower concentration due to their surface area. But then again, we don't know how they're going to work in the food chain and react in the food chain and eventually get into our food products. So I guess the point is that there's a lot of application and there's a lot of benefit, but the risks still have to be evaluated.
So you see that these nanoparticles are ubiquitous. They are everywhere in industrial society. Certainly, if you look to the right and upper right of that inner circle just outside the nanoparticle itself, biomedical and healthcare, we are extremely interested from a biomedical standpoint. And if you go to the upper right of biomedical, you see drug delivery. So in animal systems already, gold nanoparticles, for example, are being used to adsorb onto their surfaces various molecules that can either carry drugs intranasally or to some other system, or also deliver cancer-fighting antibodies or other types of drugs to them. Very complicated slide, but it gives you a lot of information and just tells you how ubiquitous these molecules really are in our daily lives. Next slide, please.
Yes.
I had mentioned that we have a collaboration with Dr. Gen Watanabe in Tokyo, and so they've been interested in the diesel exhaust particles, for example. Here we have an example of an automobile and how nanoparticles can easily be introduced into the environment. And the most common way is through engines that use gas combustion. And so they will convert gas to carbon nanoparticles in the air, and so we're going to be exposed to them on a daily basis, any areas that have motorized vehicles that use gas combustion. Also, washing off sunscreens and cosmetics. Many people everywhere in the world use sunscreens, and many of these sunscreens contain various types of oxides, like titanium, that Jeremy had mentioned, and other types of oxides.
And you have electronic fuel cells and for a renewable source energy. A lot of these products that are manufactured, there's a waste product that that can get into the wastewater, that can get into our drinking water, and can have toxicological effects. So there's research that needs to be done to see how the nanoparticles move within the environment, how they're dispersed within the environment, how they ultimately can result in new exposure paradigms.
Next slide, please.
Yes.
So the properties that make the nanoparticles toxic, there's a high level of nanoparticles reaching the environment through car exhaust. And this can be the diesel exhaust particles as well, mainly organic molecules, used in sunscreens, cosmetics, manufacturing and shipping and handling of processed nanoparticles. There's nanoparticles in our food, in our drink. They're ubiquitous within the environment. High number of particle per unit area when these particles interact with cells and the organelles, meaning that there's due to their increased surface area, they tend to have a higher reactivity than their bulk counterpart. So the high surface area ratio makes them prime candidates for the transcytosis across the cell membrane. So that reactivity due to that surface area can be exploited in order to help the particle enter the cell.
Next slide, from the last few years of some of the gold nanoparticles that we are looking at, and this is under electron microscopy. So you will see that the bar at the upper left is 1 micrometer, and the particles are on the order of 10 nanometers or less. But you do see that there is a fair amount of agglomeration or clumping of these nanoparticles. And sometimes they will clump outside cells or they will clump inside cells.
And the clumping or the agglomeration can impact the behavior of the particle within the cell. It can inhibit its uptake. It can be a factor that can also modulate its reactivity. Usually, typically, the larger the agglomerate, the decrease in surface area, therefore the decrease in the reactivity of the nanoparticle agglomerate, therefore can affect its efficacy and or its toxicity.
So you can see that we are typically not looking at a homogeneous solution of gold nanoparticles, especially once they enter physiologic fluids or culture medium or within body cells and tissues, that we start to have this clumping or agglomeration phenomenon, and it is therefore quite heterogeneous in places. Next slide, please.
The nanoparticles moving into the cell, they can translocate from distant areas within the body. And typically, they can travel wherever blood goes, similar to hormones. Researchers have concluded that the mechanism for the translocation to distant cells of the body is through receptor-mediated endocytosis. So they approach the cell, they get engulfed by the cell, and then they are taken within the cell in order to have impacts on the metabolism and the physiology of that cell. Receptor-mediated endocytosis is a good model for the nanoparticle introduction into cells because they can be spontaneously coated by proteins in the form of protein corona. And that protein corona can then dictate how it behaves in the cell or how it behaves in physiological media or blood and plasma. So that can help them act like ligands and attach the coated pits in the membranes of the cells. And I'd also like to note that different surface chemistries will attract the different suite of proteins that can then confer a different response within that cell or within a particular physiological situation. So the surface chemistry really dictates what proteins are attracted to the surface of the particle.
Your viewers and listeners will be familiar with biological mechanisms like phagocytosis or cell eating and pinocytosis, cell drinking. This is a third type of category, receptor-mediated endocytosis, where the materials can be brought in, sometimes ligands, on receptors into what are called clathrin-coated pits, and then internalized, and then they can be moved to different locations within the cell. In some cases can be degraded or recycled to the surface. There's different things that can be done. But so it appears that nanoparticles work through some form of a receptor-mediated endocytosis. Next slide, please.
Yes.
So the model that we use, remember we are reproductive biologists and doing reproductive toxicology work. So we use various cells in the reproductive system, and my work has been mainly in female reproductive function. So we work in the ovaries, and Ms. Rosemary Weber-Stelzer, the former graduate student before Jeremy, was using granulosa cells, the ovarian cells in the ovary that make up most of the ovarian follicle and that can produce the ovarian steroids like estrogen and progesterone and androgens. So what she did was to take granulosa cells and put them in vitro culture and then look at some of the phenomena that we have been telling you about. So we saw that, or we believe that receptor-mediated endocytosis can be a way in which these nanoparticles cross the membranes of these kinds of cells, the granulosa cells, and then they could also presumably move then into other organelles within those granulosa cells and achieve an effect. Some of those organelles, as I mentioned at the beginning, have to do with steroidogenesis, the process of steroid synthesis. And so it's possible that steroid synthesis could be affected and potentially, ultimately, fertility, possibly in some species, by having these nanoparticles enter the cells. And Jeremy has certainly done a lot of literature review, working on his master's thesis, and he has published several papers, like the one I think you are familiar with in nanotoxicology. He has shown that there are very few studies that have been done on toxicology with nanoparticles.
Yep. To focus on the nanotoxicology and reproduction in general is an emerging field. There's not a lot of research done. It's first coming to light. Most of the research today is focused on pregnancy, focused on male reproduction, focused on spermatogenesis and testosterone production. Most of the models in the literature are focused on male reproduction, and the female reproduction realm is just focused in pregnancy and not really on action in the ovary. So there's a great need for research focused on the ovary in order to help elucidate the mechanisms and how these particles can impact fertility.
Next slide, please.
Yes.
Okay, these are electron micrographs of granulosa cells. So we have one cell. It is a combination of four of these profiles here that you see. And at the upper left, you see a granulosa cell that is incubated without any gold nanoparticles at all. And what you see where the letter A is the nucleus, and then you will see several arrows, and those arrows are pointing to mitochondria. You can see the cristae within the mitochondria under this electron micrograph. And these are some of the organelles that are involved in steroidogenesis. If you push the next slide, you should see a yellow inset. If you click next slide,
Yes.
you will see the arrows that are within. Those are pointing to mitochondria. If you push next slide two more times, you will see there is at the upper right hand of this panel, you will see granulosa cell that is incubated with gold nanoparticles. And you will see that there are various arrows that are shown there. And those arrows are pointing to gold nanoparticles again. And the letter B there is also a single mitochondrion. And the double arrow is actually a gap junction between cells. If you now push the slides two more times, you will be in the lower left-hand panel. And what you will see there, this is panel C now, will show you letter A again, which is going to be a lipid droplet.
Interior.
And then letter B is an undamaged mitochondrion. There's another lipid droplet that you will see, but you will see little arrows, and they point to gold nanoparticles within the lipid droplet A, and also at or within the mitochondrion in B. So these are granulosa cells now separately from the rat that were incubated for 5 hours, for example, with these gold nanoparticles at a particular concentration. If you push again the slide, you will see the letter A, the lipid droplet, showing you another series of nanoparticles there. Push it two more times, you will be in the lower right-hand panel, and you will see several letters there. You will see the upper left will show you 1 micron as a as a marker, and you will see that you have undamaged mitochondria, letters B, but then if you push 2 more times, you will see damaged mitochondria. These are granulosa cells that are incubated for 24 hours with these gold nanoparticles at a very specific concentration. So the cristae are damaged, the mitochondria are swollen, and this is where one of the initial phases of steroid synthesis occurs, the rate-limiting step, which is side chain cleavage, and if your listeners and viewers are familiar with star protein or TSPO, transporter protein, this is where cholesterol is acted on at the inner mitochondrial membrane by side chain cleavage, the encoded gene product of cytochrome P450 11A1 gene. And if that is affected, that will affect the rest of steroidogenesis. So if you can click 2 more times, you will go to the next slide. And this is a graph of 4 panels again. The slide is modulation of estrogen accumulated in rat granulosa cell culture with a certain number of gold nanoparticles, 2.85 times 10 to the 10th, so a very large number of gold nanoparticles per mil. And what you see is that the incubations have been with 250,000 granulosa cells. So if you look at the upper left panel, you will see that there is actually an increase in estrogen by the cells with the gold nanoparticles at 1 hour, upper right-hand panel 3 hours, lower left-hand panel 5 hours. So there appeared to have been an effect in turning on steroidogenesis, which might have affected various enzymes that are found in the steroidogenic pathway. But if you look conversely at the lower right-hand panel, you will see that at 24 hours of incubation, in culture, rat granulosa cells exposed to 2.85 times 10 to the 10th gold nanoparticles per mil actually have a great reduction in the number or in the amount of estrogen that is produced. So whereas in the earlier panels enzymes may have been activated or possibly the membranes have been altered and they just allow more estrogen to efflux or to leave, what happens in that last panel, the lower right-hand panel, might be that the entire mitochondria are destroyed and that there are major effects elsewhere in the cell that no longer allows any more estrogen to be synthesized, and the estrogen that was accumulating in the cell has now all been released. Next slide, please.
Yes.
Okay. Now this slide here shows my experimental paradigm for my thesis work. We used 21-day-old rats, female rats, Sprague Galley rats. I injected them with equine chorionic gonadotropin in order to induce puberty, induce ovary maturation. Then after 72 hours of post-injection, I surgically removed each ovary from each animal, and one ovary from each animal was considered control. The other contralateral ovary was deemed treated. So it was a paired paradigm in which the control and the treated ovary originated from the same animal. Then the control was exposed to a vehicle control of culture medium having all supplements except for the nanoparticles. And then the treated ovary was exposed to a various specific concentration of gold nanoparticles. From there, the paired system with the control and the treated ovary remained paired through the following analyses and experiments. So you had the multiple reference gene qPCR. This is a approach that I developed using multiple reference genes to evaluate the expression of genes related to steroid biosynthesis. And I can talk a little bit later about why we use the multi-reference genes, but overall, our approach was to decrease the biological variation as well as the technical variation in order to better increase the confidence in our qPCR data, in order to evaluate potentially subtle effects in gene expression upon exposure to gold nanoparticles. We also did radium immunoassay in order to evaluate the hormone concentration or accumulation ex vivo and be able to compare and evaluate gene expression as a function of hormone accumulation. Thirdly, due to the inclusion of such low exposure doses, we had to exploit computational nanotoxicology using a computational model to predict the amount of particles that were quote-unquote seen by the ovary in order to estimate an actual target tissue dose, meaning the amount of particles that actually made contact with or were exposed to the actual tissue ex vivo. And using these three approaches in tandem, collectively, we sought to evaluate the individual variability of exposure from inter-animal differences. Ex vivo, as just clarified, I'm using the term ex vivo, meaning that the organ is intact. If the ovaries were removed from the animal, fat and excess tissue were trimmed. Ex vivo meaning that it was a whole organ culture system. Next slide, please.
Yes.
Okay. These are two electron micrographs that present how we approach some of our characterization of our nanoparticles. On the left is your typical transmission electron micrograph showing the dispersal of the size dispersal and shows the absence of agglomeration. There's some maybe minor agglomeration, but overall they're largely singlets and non-agglomerated. On the right panel, that is a micrograph that shows high resolution trans-electron micrograph. That is a very unique technique that allows us to really examine the surface structure, the crystalline geometry, to look at the molecular structure of the particles to get an idea of shape and relative structure. And the values there on the right, they correspond with the interplanar distance within the crystal structure that compose the nanoparticles. Specifically the gold nanoparticles. Next slide, please.
Yes.
Okay, I'll say a few things here. I have been talking, and Jeremy also, about the steroid biosynthetic pathway. So this is, for your viewers, steroidogenesis, or the ability of cells to synthesize these various steroids. So some of these steroids will be synthesized in the ovary, testis, in the adrenal gland, in skin even, in brain, and elsewhere. So what you will see is that in the system here, it starts with the 27-carbon cholesterol, which is then moved through kind of a tunnel, a protein called STAR, steroid acute regulatory protein, and then there's an additional cholesterol binding protein called TSPO, a translocating protein, or a PBR, peripheral benzodiazepine receptor, and they essentially will allow the movement of the cholesterol to the inner side or aspect of the mitochondrion inside the steroidogenic cell, like the granulosa cell that we're talking about, and residing at the inner membrane is the gene product for CYP11A1. That is called side chain cleavage enzyme. And it will cause 3 conversions of the cholesterol molecule and ultimately will cleave the 27-carbon cholesterol to a 21-carbon pregnenolone molecule. Now, the pregnenolone molecule goes through a particular vertical pathway called the delta-5 pathway, and then can also be converted across to the delta-4 pathway. So there's an enzyme called HSD3-beta-1, which is 3-beta hydroxysteroid dehydrogenase, which converts or metabolizes pregnenolone to progesterone, or 17-alpha hydroxypregnenolone to 17-alpha hydroxyprogesterone, or also dehydroepiandrosterone, DHEA, the first androgen in the pathway, to the most potent androgen in the female mammal called androstenedione, or A4. This androgen then is converted by another very important enzyme product called aromatase. The gene is CYP19A1 and converts androgens to estrogen like androstenedione to estradiol 17-beta, E2. And then this estrogen coming from the dominant follicles of a female mammal comprises about 95% of all of the estrogen that circulates in the bloodstream of that animal. If you look up at the upper left-hand corner again, that CYP11A1 is, as I mentioned before, the rate-limiting step of the process. If you inhibit or somehow alter that rate-limiting step, you will then alter downstream secretion of other molecules like androstenedione and ultimately estradiol and even progesterone, which is upstream of those other two but downstream from pregnenolone. Next slide, please.
Okay.
All right, this slide here shows our major genes of interest. So from the steroidogenesis pathway that Reinhold just described, we looked at genes such as STAR, CYP11A1, CYP17A1, HSD3-beta-1, and CYP19A1. So these are the genes that Reinhold just described that encode the enzymes and the transporter protein STAR that are essential to the steroidogenesis pathway. We also looked at oxidative stress via the biomarker SOD2, which is manganese superoxide dismutase, because research has shown that oxidative stress can inhibit steroidogenesis. Also, oxidative stress has been a well-documented outcome of nanoparticle exposure. Therefore, our idea was, we hypothesized that nanoparticle exposure would interact, our gold nanoparticles would interact with the mitochondrion that was shown in the work of Rose Weber-Stelzer, and that interaction would cause oxidative stress, which then would inhibit the steroidogenesis pathway. Now, we also, the reference genes that we used were GAPDH, glyceraldehyde 3-phosphate dehydrogenase, brain zinc finger protein with the symbols ZC3H15, and then beta-actin, which is a very common reference gene. We use them collectively as a suite of normalization. We use them as a suite of normalization, like I alluded to before, to help decrease the technical variation and biological variation in expression of the reference genes upon exposure to gold nanoparticles. So effectively, we took the geometric mean of the expression of the reference genes and normalized as a normalizer to effectively normalize our target genes to that collective value. Next slide, please.
Okay.
And this slide here shows linear regression analysis comparing progesterone accumulation as a function of gene expression, specifically CYP11A1A. And we used the linear regression models approach in order to look for subtle effects in the relationship between gene expression and hormone accumulation. Previous analyses, we used the two-way ANOVA to evaluate robust effects and impacts of the gold nanoparticles on both hormone accumulation and gene expression, and those robust analyses did not show any significant differences from treatment compared to control. Therefore, we took it a step further to look for more subtle effects using these regression models in order to tease out any potential effects that might be shadowed or hidden by the more robust two-way ANOVA approach. And we can see here that there were a subtle effect, a subtle increase in a positive relationship between progesterone accumulation and the expression of CYP11A1.
So as Jeremy had mentioned, there are subtle differences here, but there appears to be a statistically significant increase in the ratio of progesterone accumulation relative to the paired control versus the relative expression of CYP11A1 that encodes side chain cleavage, the rate-limiting step of steroidogenesis. If you look at the upper left of that figure, you will see that there are 30 rats. It's highly significant, p less than 0.001, and the R squared, or the coefficient of determination, is 0.366. And that essentially tells us that about 36 or 37% of the effect that we're seeing is due to the nanoparticles here. But that also means, even though this was significant, that leaves an additional 64 or so, 63% that may be due to some other considerations, not just the gold nanoparticles.
You know, and I'd like to also add that this kind of representation shows us the variability in response in individual animals, meaning that there's an individual differences between the animals that may play a role in the observed effect.
Yes?
Next one. Okay, so what I put in here, these I can hopefully go through relatively quickly. We've used different model systems. We've used rat model systems. We've used in vitro, ex vivo. Jeremy also did a study where we were using the sea urchin as a model system, looking at environmental exposure, since even in water you would have many of these different nanoparticles. So we used the purple sea urchin, Strongylocentrotus purpuratus, as a model. So if you go to the next slide, this is phase contrast interference microscopy looking at two-cell sea urchins after in vitro fertilization. Takes about 90 minutes approximately to split to two-cell. You can see the size. There's a 50 micrometer marker at the bottom. If you go to the next slide, you will see that at about 4 hours they begin to split to 4-cell and 8-cell stage. You go to the next slide.
Yeah.
This is a fixed topically stained with hematoxylin and eosin blastula stage after 24 hours. And then if you go to the next slide, and I don't know if your viewers will be able to see this, but this is now normal microscopy. If you click on it, you might be able at the bottom to see that they will move. Are they moving for you? If you click at the arrow, this just shows you blastulas and ciliary motion, allowing them to move in circles at 24 hours. If you go to the next slide, you will see the final stage, and we evaluated these various stages, Jeremy did. This is called the pluteus stage and can be seen anywhere from 2 to 5 days, depending on culture. So what Jeremy evaluated was, do the gold nanoparticles on the next slide inhibit sea urchin development from 2-cell to the pluteus stage? So what you see here is the mean percentage of these cells going from 2-cell to pluteus over 48 hours. The control, we have about 55%. And then with looking at any of the 3 concentrations of gold nanoparticles, 2.85 times 10 to the 4th, 28 million, 2.85 times 10 to the 10th, you will see that the 2.85, 10 to the 4th, and to the 10th are significantly reduced from the control, whereas the middle blue one was not. But it showed us that although there were no effects of the gold nanoparticles on cells going to 2-cell or 4-cell or some of the other stages, it did reduce significantly the ability of the cells to go from 2-cell to this last stage, pluteus, at 48 hours.
And I'd like to also add that there was, this was a relatively small sample size. I think that if the experiment was repeated several more times, that middle dose might show some significance.
If you can go to the next slide, one of the last slides we have what we're calling our take-home messages from the 2 major studies that we talked about, Rose Weber-Stelzer's and Jeremy Larson's. From Rose's study, we showed that 10 nanometer gold nanoparticles can definitely enter granulosa cells as visualized by electron microscopy, and they appear to significantly reduce the accumulation of estradiol at 24 hours, but also increasing the estradiol release or synthesis at 1 hour, 3 hours, and 5 hours. And then using the model that Jeremy developed, he showed that these same gold nanoparticles can affect the expression of several of the genes involved in steroidogenesis, including STAR, CYP11A1, and hydroxysteroid dehydrogenase. The last study that we just showed you, gold nanoparticles can inhibit sea urchin development to the pluteus stage in the laboratory setting. If you go to the next slide, it shows you that while the biochemical effect can differ with respect to toxicant, because we've looked also at dioxins and many other molecules, it's very important to look at, and they can be different due to dose of administration, how long you administer, the duration, the animal model, the model system. Even though all those things can be different, we think that the changes that we see, the modulation or the inhibition in some cases of estrogen synthesis, may play a role in the reduced fertility that's seen in animals and potentially in humans that are exposed to some of these molecules. Certainly we've seen it with dioxins, we're seeing it with xenoestrogens, and it's possible that we'll see something similar with gold nanoparticles that are found in the environment. The final slide then is just showing the funding sources that we've used. The National Institutes of Health in Washington, D.C., in the United States of America, has supported this, especially from the National Institute of Environmental Health Sciences, and also the National Institute of Child Health and Human Development, and some of their subdivisions. Thank you very much. Obrigado. That was the entire slide presentation, Paulo.
Okay. Reinhold and Larson, thank you so much for the presentation. And very good. And I would like to see if my proposal will be good for you, because here is 12 o'clock, okay?
Yes, okay.
And he doesn't have time to discuss this very good presentation.
Sure, we have time. If
Our program is 11 to 12. I told you in the beginning that 1 hour in the beginning is so long, but in the end is not.
You were right. You were correct, Paulo.
Pois é, yes.
You were right.
Yes, but I would like to make now one proposal. Maybe he can have another program.
When would you like to do that?
To discuss this thing that you show for us.
Yes.
It's very important, this kind of research. And
when would you like to do that? Now or?
no, in another program.
Okay, that would be fine, Paulo.
Maybe April 20.
Okay, you and I can be in communication through Skype text, and we can come up with another date.
Okay, very good.
Very good.
Thank you very much.
Because, one moment. Because this kind of research that you and Larson is doing
Yes,
is not common.
right.
It's not
It is not
common. And here in Brazil, he doesn't have this.
Yes.
And
I understand.
and it doesn't have here because the public money that he have is for new knowledges in science to production new products.
Yes, I understand.
Is not for science to produce knowledge for impacts the nanotechnology.
Yes, I mean, that really is what truly is happening everywhere, that very much money is going in. I think it was said to be $1 trillion industry by 2016 or this year already. So there's very much money going into, as you say, the production of the nanoparticles, characterizations, things like that, but very, very little looking at the possible toxicologic and other biologic effects.
Yeah. So when he have any kind of research like yours, I think is very, very important. We have another opportunity to discuss this.
That would be fine. We would be glad to do that, Paulo.
Okay. Então, I will send you new email to discuss a new opportunity for discuss your research.
Very good.
Very good.
Thank you very much. Obrigado. Thank you.
Obrigado você por colocar tudo isso, todas essas coisas importantes, não é, para a gente.
Tchau.
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 Até.